First, I would like to thank Ernst for organizing and also for this opportunity to talk to you about some work that we do in our group. So just to give you a bit of a broader sense of what we do, as my topic is a little bit the same and also at the same time different from the talks that we have heard so far, our group is very much interested in understanding bacteria surface interactions and also finding ways to control that bacterial adhesion and hopefully down the road learn how to interfere with biofilm formation. Biofilm being a not so easy problem to tackle. And so with that goal in mind, research in our group is actually divided into 4 major themes. So as I mentioned, we look at bacteria surface interactions. So here we focus on initial attachment of bacteria to surfaces. And then we are also interested in measuring those interaction forces at the single molecule and single cell levels via the atomic force microscope or AFM. And also we, in the group, we develop analytical tools that will help us measure physical properties of biofilms. So in this research area here, quantitative imaging of biofilms. So analytical tool that can measure biofilms with high spatial resolution as well as under physiological conditions. And somehow complementing these 3 aspects of our work, we are in parallel developing nanomaterial-based antimicrobial. And as you can easily guess, in today's talk, I would be focusing mostly in our nanomaterial-based antimicrobial development. So this antimicrobial is meant to target bacteria and biofilms. So there are different approaches or different ways that one could target bacteria or biofilm infections. So I'm just going to show you the popular ones. So one of them is surface modification. So as the approach implies, we tune physical properties of the surface in the hopes that we could either suppress attachment of bacteria or even kill them upon contact. And another approach is physical removal of biofilms. So in here, the premise is to disrupt the biofilm structure. And you can actually do it via different ways. So it could be mechanical, it could be light-based, it could also be sound-based. And the third one, which is, I believe, very close to the crowd here, is the use of nanoparticles and small molecule agents. So just to put this into context, it's great that the intro we heard from Urs at the start of this conference. He gave a very good overview of the functionalities and advantages of using nanoparticles. But in the context of the bacteria, we want our nanoparticles to destabilize the membrane. And if we designed it accordingly, we could also penetrate across the biofilm depths or biofilm crevices. And in the field, in at least biofilm area, we wanted to target this, what we call as the persister cells, which are mostly found closer to the bottom or at least inside the biofilm. And so what I would like to talk to you today is our approach in designing a nanoparticle-based antimicrobial with multiple functionalities, which leads me to the title of my talk, Smart Nanomaterials for Multi-Pronged Strategies Against Bacteria. So this is actually a short version of a much longer title, Multifunctional and Stimuli-Responsive Polydopamine Nanoparticle or PDNP-based platform for targeted antimicrobial applications. So this is the focus of my talk today, and I promise that I would try to parse out the many keywords that you see in this title here. So by the way, this is primarily the work of Dr. Nesha Andoy in the group. So first, let us refresh ourselves with the properties of a suitable nanomaterial-based antimicrobial platform. So we've heard it many times, or at least more than once in the conference today, but I would just like to put it into context in bacterial field and also a biofilm. So clearly, we want our our platform to be biocompatible, meaning it could attack the pathogen, but spares the healthy mammalian cells. And also, we want it to be stimuli responsive so that we have control over the onset of bactericidal action. For example, when we want to release the drugs. The other property, which I would say very loaded, is we have these multiple antimicrobial mechanisms or functionalities. So this actually reminds me of what our previous speaker talked about, Miffy Cheng from U of T. This is similar to the all-in-one, one-for-all platform. But what I mean here is that we have this platform who can kill and also a platform platform that can act as a nanocarrier, a platform where there is a facile surface functionalization, so we could confer it with extra functionalities. So one of them clearly is specificity, and also we want it to be flexible. So like those smart polymers who could kill bacteria, but at the same time release them once dead. So in our group, to address the biocompatibility property, we start with this bio-inspired polydopamine nanoparticle, or simply PDNP. This is bio-inspired, dopamine being a neurotransmitter that is already found inside the body. So we like working with this PDNP because it is so easy to synthesize in the lab. So basically, you just throw in dopamine monomers under basic condition, and then within an hour or so, you generate your nanoparticles. And so we did monitor how these nanoparticles grow through time. And within an hour or so, you can already get nanoparticles of about 45 nanometers in diameter. And so to visualize these nanoparticles, we also look at its structure via TEM and also AFM. And what I'd like to highlight here is that based on these images, we are confident that we are able to synthesize nanoparticles in our ideal size range. And that is less than 100 nanometer, so meaning smaller than the dimensions of the bacteria. And so now that we have synthesized and characterized our nanoparticle or our PDNP, the next thing we check, and I didn't mention this to you yet, the next thing we check is its photothermal capability. So people who started working with PDNP, they have already shown that PDNP heats up when shown by laser. And so in our group, we just monitor the solution temperature as we increase the concentrations of PDNP. And we did so via a thermal imager. And here's what we got. So as expected, with increasing concentrations of PDNP, we actually also increases the temperature right here. And what I'd like to draw your attention to is that if you look at the 1 nanomolar nanoparticle concentration, it actually corresponds to heating or temperature that is close to 50 degrees Celsius. So that's good. We were able to show the photothermal capability of PDNP. And of course, obviously, we tested its laser-induced heating efficiency in killing a drug-resistant E. coli. So here is a plot of bacterial growth as a function of the nanoparticle concentration. And what you can see here is that bacteria do die with increasing PDNP, or you increase the viability with increasing PDNP concentration. And so if I fit this data into what we call an M-Gompertz function to determine its MIC, or minimum inhibitory concentration, we get an MIC that is 1 nanomolar. So this is actually well and good. This is good news. It just shows that we can use this photothermal capability of PDNP to kill a drug-resistant E. coli in a stimuli-responsive manner. But then it caught us really quickly that if I go back to the solution heating temperature that I showed you earlier, 1 nanomolar nanoparticle concentration actually corresponds to heating happening at 50 degrees, which affects not only the bacteria or the pathogen, but also the surrounding healthy cells. And in fact, I'm pretty sure many of the audience in the crowd could actually tell me that we do have a small window, or a very narrow window of opportunity when we want to do photothermal heating, as it has been shown in this very nice work here published in Science in 2017. It only takes upwards of 42 degrees Celsius to kill human cells, and upwards of 47 degrees Celsius to kill E. coli. So we have to make sure that we could operate in this very narrow window of opportunity. But many people here know that this is actually not new, and this is in fact a big challenge in the field. And so one way to mitigate this is to specifically target the target. So in our case, specifically target the bacteria. So we tried this approach, and the way we did it is starting with our bare polydopamine nanoparticle, we further decorated it with an antimicrobial peptide with dual functionality. And that dual functionality is that, number one, it could target bacteria, which just means it is more selective to bacterial membrane than mammalian cell. And second, because it's an antimicrobial peptide, it has its own intrinsic bactericidal or lytic activity. And add to that, the photothermal treatment that is inherent in PDNP, we have laser activated killing in addition. So what we ended up in this design or in this platform is multiple and coordinated biocidal mechanisms that could target pathogens. So this sounds like a good concept, but of course you have to make sure if you could indeed immobilize this peptide onto our nanoparticle surface. So here I am showing you once again AFM images of the bare nanoparticle to your left, and also in the presence of the peptide to your right. So I am not sure how easy it is to see from your screen, but if you actually look closer, you could see some nanoscale protrusions in the presence of the peptide. And we've done some analysis on this, and we did see an increase about 5 nanometer in the diameter of this nanoparticle in the presence of peptide. But if you are not convinced, here is our zeta potential. So for bare PDNP, we have this negative charge, and there is a dramatic shift from negative to positive in the presence of peptide. So what this slide basically shows you is that we are now confident that we have successfully immobilized this AMP onto our nanoparticle surface. But that's not the end of our consistency checks. The next thing we have to make sure is that can it target bacteria? And so the way we did it is we incubated this, first, we incubated bare nanoparticles with bacteria, so no presence of peptide yet. And I'd like to, so this, by the way, are AFM images. So this is your bacteria. And what I'd like to draw your attention to is inside the white circles here. So what you see here are the nanoparticles or clusters of them. And what this tells you is that for bare nanoparticles, so meaning no peptide, you actually see more of these nanoparticles outside the bacteria than on the membrane. And when we add peptide into the mix, so meaning we incubate the bacteria on PDNP that is decorated with a peptide, these are the images we got. So very clearly, the clusters of nanoparticles have now more affinity towards the bacterial membrane, nicely demonstrating that we do have some efficient targeting. So we are happy with this result. And the other important thing that we check is, can the peptide still retain its lytic activity even after immobilization? And so that's what we did. We tried to grow it, the bacteria in the presence of this platform, PDNP and peptide, and we do still retain its lytic activity. So now that we have this, so now that we are sure that we have a platform that could target the bacteria, that could still retain its activity, and this laser-induced heating, we of course next tried how efficient it is in killing a drug-resistant E. coli. So here again is a plot of bacterial viability as a function of this peptide nanoparticle concentration in the absence of the laser, so the green curve here, and in the presence of the laser, this red curve here. So what you see here is that in the presence of the peptide, you do see a slight decrease in MIC, which means that there is some synergistic effects that we could already say from the action of both the peptide and the laser. But perhaps a more compelling comparison is when I compare the system or this platform to just bare PDNP in the presence of the laser. So here you see this gray curve. This is just your bare nanoparticle in the presence of laser, and the red curve here is your nanoparticle plus peptide in the presence of the laser. And so when we fit it to the M-Gompertz function, we are, even if you just take a visual look here, you can see that there is a slight improvement in the MIC. And to the skeptics in the audience, you may say like, oh, the improvement is actually just very slight. But I am not done yet. What I haven't showed you yet is the MIC that corresponds to this red curve here is actually close to 0.4.5 nanomolar. So when I bring up the solution heating temperature, the MIC or the concentration of the nanoparticle that we're getting here corresponds to heating happening at a much, much lower temperature than 50 degrees Celsius that we previously observed. So we are now killing bacteria, not following the general hyperthermia higher heat, but then we are killing it at a much lower temperature of 40 to 42 degrees Celsius. So what we think is happening here is that in the presence of the peptide, bacteria is brought in close proximity, or sorry, the nanoparticle is brought in close proximity to the bacteria. And once it's in contact with the bacteria, it starts to do its lytic action, which is because it's an antimicrobial peptide. So it already starts to introduce membrane damage. Now, when we add laser in addition, we induce further membrane damage. So I'd like to describe it as some kind of a bacteria experiencing a double whammy effect, or it's like a double jeopardy. And it does so via this mechanism we call as localized nanoheating. And so to test that hypothesis, we compared the topography of bacterial membrane without anything, bacterial membrane that are incubated with the nanoparticle in the presence of the peptide. So you've seen this already, it attaches to the membrane. But then when we add the laser in addition, we observe this membrane ruffling, which is a very good indication of membrane being damaged. And so we tried to quantify this damage on the bacterial membrane or bacterial cell envelope via Young's modulus determination. So Young's modulus is just one metric that we use to measure how resistant is a material to deformation. So higher value means more resistant to deformation. So here is a plot of that Young's modulus as a function of the different conditions. So for our control bacteria, meaning grown happily in the lab, no constraints or no nanoparticle or no peptide and no laser, it has a Young's modulus of about 9-10 megapascal. In the presence of the laser, we brought it down to about 4 or 5 megapascal. Sorry, this is without laser, but in the presence of the peptide. Now, if we add laser in addition, we bring it down much lower. And so we thought this is a very nice indication of membrane damage that highlights the synergistic effects of photothermal treatment and antimicrobial peptide in a single nanoparticle-based platform. So with that, I hope I have convinced you that combination of this photothermal heating and the stabilizing activity of an antimicrobial peptide actually gives us, or at least a starting platform that is biocompatible, similar responsive, and capable of multiple killing mechanisms. So in the few minutes that I have, oh, by the way, we are also looking at another nanomaterial in our group, serotonin, so instead of dopamine, which is serotonin is a close cousin of dopamine. Nothing much is known about serotonin yet. So we are generating polyserotonin nanoparticles, and we are still, so we just finished one work in which we were able to find the condition to generate nanoparticles in our ideal size range. So when I say again, ideal, about 100 or less than 100 nanometer. So with that, I'm just gonna quickly show you the one aspect that we do that we are very excited about, quantitative imaging of biofilm. So this is an analytical tool that we try to develop to help us measure properties of biofilm. And we actually like this because we like this because we wanted to use it as a way to also quantify the effect of our engineered nanomaterial-based antimicrobial. So far, these are nice images that we can now look at biofilm topography under physiological conditions, where you can see here the biofilm, the bacteria inside biofilm, and the network here, which we believe could be EPS, and then correlate that to layer thickness, so the extracellular polymeric substances thickness, and also the elasticity or the Young's modulus of the biofilm interface. And I would like to highlight this, that imaging biofilms in native state is actually not very easy. So this is one thing that the group is really excited about. And so we wanted to use the tool to now add our nanoparticle into biofilm and then do some more physical and mechanical characterization. So very lastly, this is my last slide. We heard about penetration as a problem in nanoparticle application. So that is also one area in the group that we just started to tackle. And this is a preliminary data showing you that when we try to incubate this bare nanoparticle, so nothing yet, no peptide, no anything, just the bare nanoparticle, we tried to incubate it in our biofilm, and then we took a cross section and then look at the image in the TEM. PM, we were able to see that this PDNP could actually penetrate the biofilm depth. So you can see here the blue dotted line is the biofilm surface. And if I measure all the way down to where I could see these PDNP nanoparticles, it's about 15, 16 microns. And what is exciting about this result is this PDNP tend to not cluster inside this biofilm. So we are really excited about it, and we are continuing the design of our nanoparticle so that we could improve this penetration. So with that, I'd like to thank the group who did all the heavy lifting, and also, of course, the funding agency. Thank you again to the organizers and to you for your attention.
Thank you, Ruby. So we have one quick question from the audience. I saw aggregation of the particles. Can you elaborate on the stability and zeta potential of the particles?
Yeah, so the zeta potential of this polydopamine nanoparticle, so the bare one is negative. In the presence of the peptide, it becomes positive, which is expected. So yes, you are very right. It aggregates, and so the what I've shown you here, these are actually clusters of nanoparticles, because the bare nanoparticle is less than 100 nanometer. But then if it's clusters of them, then you can see this bigger size here. But that being said, though, it doesn't stop the photothermal capability of the PDNP or the photothermal action of PDNP. So they tend to cluster, but then in terms of executing its photothermal action, we could still get it. But yes, you are right. We do get aggregation.
Okay, thank you, Ruby. So we still have a couple more questions from the audience, but maybe you can answer them online.